Hello there, and welcome to Sleepy Science Vault. What does it actually mean for something to grow old? It's a question that seems simple on the surface, but biologists have spent decades trying to answer it with any real precision. And what they've found is far more intricate than most of us imagine. In the language of biology, aging is generally described as a gradual loss of physiological integrity over time. It's not a single event, and it's not a switch that flips on a particular birthday. Instead, it's a slow accumulation of small changes that, bit by bit, reduce how well our bodies function, and gradually increase the likelihood of disease, and eventually, of death. But here's where things become genuinely fascinating. The number of candles on your birthday cake, your chronological age, is not the same thing as your biological age. Chronological age simply counts the years you've been alive. Biological age, on the other hand, reflects something deeper. It reflects how old your cells and tissues actually behave, how worn, or how resilient, the inner machinery of your body truly is. Two people can be exactly 40 years old on paper, and yet, biologically, one might resemble someone in their early 30s, while the other carries the cellular signatures of someone closer to 50. Same number of years. Completely different internal stories. So how do scientists actually measure something as abstract as biological age? One of the most remarkable tools developed for this purpose is known as the epigenetic clock. To understand what that means, it helps to picture your DNA not just as a string of genetic instructions, but as a long manuscript covered in tiny annotations, small chemical marks that influence which genes are turned on, and which are kept quiet. These marks are called epigenetic modifications, and one of the most studied types involves a process called DNA methylation. What scientists discovered was startling. These methylation patterns don't just shift randomly over a lifetime. They change in remarkably predictable ways, almost like a slow, steady rhythm written into the body itself. By examining these patterns, researchers can estimate a person's age with extraordinary precision. In fact, DNA methylation can predict chronological age with up to 96% accuracy. Just from a small sample of cells, an epigenetic clock can come remarkably close to guessing how many years someone has actually lived. But the deeper value of these clocks isn't just predicting age, it's revealing something more personal. Because sometimes, the epigenetic clock doesn't match the calendar at all. For some people, the clock runs faster than their actual age, suggesting that, biologically, their body is aging more quickly than expected. For others, it runs slower, hinting at a kind of internal resilience, a body that seems to be aging more gently than the years would suggest. And this single observation opens a door into something profound. Aging isn't just about time passing. It's about how the body responds to that time. Now, you might imagine aging as something smooth, a slow, steady decline, like sand falling gently through an hourglass. But at the molecular level, that's not quite what happens. Aging is not perfectly linear. Instead, research has shown that many biological markers shift in noticeable waves, with two particularly dramatic turning points, one around the mid-40s, and another around the early 60s. During these windows, certain molecules in the blood, certain proteins, certain metabolic signals, seem to shift more rapidly than at other times in life. It's almost as if the body moves through distinct chapters, rather than a single uninterrupted story. And perhaps most strikingly of all, aging is profoundly different from person to person. Two individuals can walk through life side by side, born in the same year, and yet age in completely different ways. Some accumulate cellular damage more slowly. Others experience certain biological shifts earlier, or more intensely. This diversity isn't random noise, it reflects real, measurable differences in how damage builds up within our cells over time. Which means aging isn't simply something that happens to us. In many ways, it's something that unfolds with us, shaped by biology, environment, and the countless small moments that make up a life. As we continue through this journey into the science of the human body, we'll explore exactly what that damage looks like, how it builds, how it can sometimes be slowed, and what it truly means to grow older, not just in years, but in the quiet, intricate language of ourselves. If you could shrink down, small enough to drift quietly through the inside of a single human cell, you would witness something remarkable. A constant, tireless effort. Repairs being made. Damage being cleaned up. Tiny machines working around the clock, simply to keep things running. This is where the real story of aging begins. Not with wrinkles, or gray hair, or the slow creak of joints, but deep within the microscopic world of our cells, where the true causes of aging quietly unfold. For a long time, people searched for a single explanation. A single switch. A single gene that, once flipped, would set the aging process in motion. But the more scientists looked, the more they realized that no such switch exists. There is no single aging gene. Instead, aging emerges from something far more complex, a web of interconnected influences. Genetics plays a role, shaping the tools each of our cells are given to begin with. Cellular damage plays a role, accumulating slowly over the years. And so do environment and lifestyle, the air we breathe, the food we eat, the stress we carry, the sleep we get, or don't get. All of these threads weave together, and from that weaving, aging emerges. It helps to think of aging not as one process, but as many processes, happening simultaneously, across nearly every system in the body. The cardiovascular system, the immune system, the nervous system, the skin, the bones, the muscles. Aging touches all of it, not all at once, and not in the same way for everyone, but gradually, and persistently, across a lifetime. To really understand why this happens, we need to travel down to the smallest scale imaginable, down to the molecules that make up our cells. At this level, aging involves damage. Damage to DNA. Damage to proteins. Damage to lipids, the fatty molecules that form the walls and structures of our cells. Each of these components plays a vital role in keeping a cell alive and functioning properly, and each of them, over time, can become worn, frayed, or broken in small ways. Now, this kind of damage isn't unusual. In fact, it's happening constantly, even in a perfectly healthy young body. What matters is what happens next. because our cells aren't helpless against this damage. They come equipped with an extraordinary set of repair systems. Molecular tools whose entire job is to detect problems and fix them, almost as soon as they appear. In a young, healthy cell, these repair systems work with astonishing efficiency. Mistakes in DNA are caught and corrected. Damaged proteins are broken down and replaced. It's a quiet, continuous maintenance process, one that mostly goes unnoticed, because it works so well. But as the years go by, these repair systems begin to slow down. They don't stop working entirely, but they become less efficient. Mistakes that would once have been caught and corrected quickly begin to linger a little longer. Damage that would once have been cleared away starts to accumulate, just slightly, day after day. And to understand just how much repair work is actually happening, consider this number. Every single day, the DNA inside each one of your cells is damaged roughly 100,000 times. It's a staggering figure. 100,000 small disruptions, per cell, per day. Most of these are minor. Most are fixed almost instantly. But across trillions of cells, repeated day after day, year after year, even small inefficiencies in repair can begin to add up into something much larger. Over time, this becomes a kind of slow, quiet accumulation of biological stress. Not a sudden event, but a gradual build-up, the kind of thing that's almost invisible in any single moment, but becomes undeniable across decades. One major contributor to this stress comes from something called oxidative stress. As cells go about their normal activities, particularly the process of generating energy, they produce certain byproducts known as reactive oxygen species, often shortened to ROS. In small amounts, these molecules are a normal part of life. But they're also highly reactive, meaning they can interact with, and damage, other molecules nearby, including DNA, proteins, and lipids. When the balance tips, when ROS accumulate faster than the body can neutralize them, oxidative stress begins to build. And over time, this contributes to the slow molecular wear and tear that defines so much of the aging process. Much of this energy production, and much of this oxidative activity, takes place inside tiny structures within our cells called mitochondria. Mitochondria are often described as the powerhouses of the cell, and for good reason. They're responsible for converting nutrients into usable energy, fueling almost everything our cells do. But like so much else in the body, mitochondria change with age. In youth, they tend to function efficiently, producing energy smoothly while keeping byproducts like ROS relatively well controlled. But as the years pass, mitochondria can become less efficient. Energy production may decline, and the balance between helpful and harmful byproducts can begin to shift. When you bring all of these threads together, the gradual decline in repair efficiency, the relentless background noise of DNA damage, the slow creep of oxidative stress, and the quiet aging of our cellular powerhouses, a clearer picture begins to form. Aging, at its core, isn't a single dramatic event. It's the slow, cumulative result of countless tiny processes, repeating, again and again, across millions of cells, for decades. And yet, within this complexity, there's also something quietly reassuring. Because if aging is shaped by so many different factors, genetics, damage, environment, lifestyle, then it also means aging isn't entirely fixed. There is room, within this intricate system, for influence, for care, for small choices that, over time, can shift the balance. In the chapters ahead, we'll continue exploring this hidden world, moving deeper into the systems of the body, and uncovering more of the quiet, intricate processes that shape the long, slow story of growing older. Picture, for a moment, a vast and quiet city, humming softly in the background of your life, every hour of every day. Power plants, generating energy. Cleaning crews, sweeping through streets and buildings. Engineers, maintaining the structures that everything else depends on. This city never sleeps. It never stops. And it is, of course, your body, at the cellular level. We left our last journey with the mitochondria, those small but mighty energy producers, gradually becoming less efficient with age. And this single change ripples outward, touching nearly everything else. When energy production declines, even slightly, the effects don't stay isolated. They begin to surface as some of the very symptoms we associate with growing older. A subtle dip in stamina. A feeling of needing a little more rest than before. Recovery that takes a touch longer than it used to. None of this happens overnight. It emerges slowly, the natural consequence of cells having a little less fuel to work with. And this connects directly to something deeper, metabolism itself. Metabolism isn't just about how quickly we burn calories. It's the entire web of chemical processes that keep our cells alive, processes for generating energy, building new molecules, breaking down old ones, and maintaining balance. And as the years pass, this web begins to shift. These metabolic changes are tightly linked to aging, not as a side effect, but as one of its central threads. The way cells produce energy, the way they respond to nutrients, the way they signal to one another, all of it gradually evolves, and these shifts help shape much of what we experience as growing older. Now, within this vast cellular city, there's one process worth pausing on for a moment, because it plays such a crucial role in keeping everything clean and functional. It's called autophagy, and the name itself is rather poetic. It comes from Greek roots meaning, quite literally, self-eating. Autophagy is the process by which cells identify their own damaged or worn-out components, and break them down, recycling the pieces into something useful again. It's a bit like a meticulous cleanup crew, moving quietly through the city, clearing away debris before it has the chance to pile up. In youth, this process tends to run smoothly. Damaged proteins, broken organelles, cellular clutter, all of it gets identified and cleared away in a steady rhythm. But as we age, autophagy tends to decline. The cleanup crews become smaller, slower, less responsive. And when that happens, debris that would once have been cleared away quickly begins to linger. Bit by bit, it accumulates. This connects closely to another concept, one with a rather elegant name, proteostasis. Proteostasis refers to the delicate balance of protein production, folding, and disposal within a cell. Proteins are remarkable molecules, tiny, intricate structures, each folded into very specific shapes that allow them to do their jobs. But sometimes, proteins misfold. Sometimes, they become damaged. And under normal conditions, cells have systems in place to detect these flawed proteins and remove them. When proteostasis begins to fail, and especially when it's paired with declining autophagy, these damaged or misfolded proteins are no longer cleared away as efficiently. Instead, they begin to accumulate, sometimes clumping together inside or around cells. It's a quiet kind of clutter, invisible to the eye, but with very real consequences for how cells function over time. Now, alongside this, there's another process worth understanding, one that involves something we encounter every day, often without realizing it: sugar. It's called glycation. Glycation happens when sugar molecules in the body attach themselves to proteins or fats, often in ways that weren't intended. Over time, these sugar-protein combinations can form structures that are stiffer, less flexible, and more resistant to being broken down. You can imagine it almost like a kind of slow, internal stiffening, tissues that were once supple and elastic gradually becoming a little more rigid. And this stiffening doesn't just affect structure, it can also encourage low-level inflammation, adding yet another layer to the body's accumulating burden over time. So far, we've explored damage to proteins, to cellular cleanup systems, to energy production. But there's another, quieter kind of change happening as well, one that takes place not within the proteins themselves, but within the very instructions that guide how cells behave. We touched on this earlier, when we explored epigenetic clocks. But now, let's look a little closer. As we age, epigenetic changes accumulate. Remember those small chemical marks that sit upon our DNA, helping to determine which genes are active, and which are kept quiet? Over time, these marks shift. Some genes that were once carefully regulated may become more active than they should be. Others may become less active than they need to be. It's not that the DNA itself is necessarily damaged, it's that the instructions layered on top of the DNA begin to drift, ever so slightly, from their original pattern. And this drift connects to something called chromatin. Chromatin is the structure formed when DNA wraps tightly around proteins, creating a kind of organized, compact packaging within the cell's nucleus. This packaging isn't just for storage, it plays an active role in controlling which genes are accessible and when. As we age, this chromatin structure can begin to loosen or become disorganized in certain regions. And remarkably, research suggests this kind of chromatin breakdown can contribute to aging on its own, independent of any actual mutations in the DNA sequence itself. In other words, sometimes, it's not the information itself that changes. It's how that information is stored, organized, and accessed. This idea becomes especially significant when we consider the brain. Some research suggests that a loss of epigenetic information, this gradual drift in the instructions layered upon our DNA, may play a meaningful role in brain aging. The way neurons access and use genetic information may shift over time, contributing to some of the changes we associate with an aging brain. And finally, let's turn to one more concept, one that researchers consider absolutely central to the aging process: cellular senescence. As cells experience damage, stress, or simply the wear of time, some of them eventually reach a point where they stop dividing altogether. They enter a kind of frozen state, no longer growing, no longer reproducing. These are known as senescent cells. But here's the curious part. Even though senescent cells stop dividing, they don't disappear. And they don't simply sit quietly in the background, either. Instead, they remain active. They continue to exist within tissues, releasing various signals and molecules into their surroundings, influencing nearby cells and the environment around them in ways that can ripple outward across the body. In many ways, senescent cells represent a kind of paradox, cells that have stopped their normal function, and yet continue to exert influence, long after their original role has ended. Taken together, reduced energy production, shifting metabolism, declining cleanup systems, accumulating protein clutter, stiffening tissues, drifting genetic instructions, and the quiet persistence of senescent cells, we begin to see aging not as a single thread, but as an intricate, interwoven tapestry. Each of these processes influences the others. Each adds, in its own small way, to the slow unfolding story of a body moving through time. And in the chapters ahead, we'll continue following this story, exploring exactly what senescent cells do once they remain in our tissues, and how the body responds to this quiet, lingering presence as the years continue to pass. When we left off, we were sitting with a strange little paradox, cells that have stopped dividing, and yet remain very much alive. Active, present, still capable of influencing the world around them. So what exactly do these senescent cells do once they settle into this strange, suspended state? It turns out, quite a lot. One of the most significant things senescent cells do is release a steady stream of signals into their surroundings. Scientists refer to this collectively as the SASP, short for the senescence-associated secretory phenotype. It's a rather technical name for something that, conceptually, is fairly easy to picture. Imagine a cell that has, in a sense, given up its original job. It no longer divides. It no longer contributes in the way it once did. But instead of fading quietly into the background, it begins broadcasting. Releasing a mixture of molecules, inflammatory signals, proteins, and other compounds, out into the surrounding tissue. At first, this might sound like a relatively isolated event. One cell, releasing a few signals nearby. But the effects don't stay contained. These signals travel. They reach neighboring cells, healthy cells, cells that are still doing their jobs, still dividing, still functioning normally. And remarkably, exposure to these signals can begin to influence those healthy cells too, sometimes nudging them toward a similar senescent state, or simply disrupting their normal behavior. In this way, dysfunction doesn't stay isolated. It spreads. Quietly, gradually, like ripples moving outward from a single point, slowly affecting a wider and wider area of tissue. And here's where the story becomes especially important. Over time, these senescent cells don't just appear and then clear away. They accumulate. In a young, healthy body, the immune system plays an active role in identifying senescent cells and removing them, a kind of ongoing quality control. But as the years pass, this clearance process becomes less efficient, much like the other repair systems we explored earlier. And so, senescent cells begin to build up, settling into tissues throughout the body. Researchers sometimes refer to them, somewhat playfully, as zombie cells. Cells that are technically alive, but no longer functioning in their intended role, lingering, persisting, continuing to release their disruptive signals, tissue after tissue, year after year. For a long time, scientists debated exactly how important these accumulating senescent cells really were. Were they simply a byproduct of aging, a kind of cellular debris left behind as an inevitable consequence of time passing? Or were they something more active, something that actually contributes to the aging process itself? The growing body of research points towards something quite striking. Senescent cells appear to be causal drivers of aging, not merely byproducts. In other words, it's not just that aging causes senescent cells to accumulate. It's that the accumulation of senescent cells actively contributes to aging itself. Their presence, and the signals they release, appear to genuinely influence how quickly tissues age, and how function declines over time. It's a subtle but profound shift in understanding. Aging isn't simply something that happens to these cells. In some ways, these cells become participants in the process, quietly shaping the experience of growing older, tissue by tissue, system by system. And with that, we begin to move from the hidden, microscopic world of cells, into something more visible, more familiar. The way aging actually shows up, in the body, in ways we can see, and feel, and notice, over the course of a lifetime. Let's begin with something many of us don't think much about, until we start to feel it, our muscles. As we age, the body gradually loses muscle mass and strength. This process has a name, sarcopenia. And here's something that often surprises people. Sarcopenia doesn't begin in old age. It doesn't wait until our 60s, or 70s, to quietly start its work. It begins around age 30. For most of us, our 30s feel like a time of stability, a plateau, of sorts, after the rapid growth of childhood and adolescence. And in many visible ways, that's true. But beneath the surface, in the slow language of muscle fibers and cellular turnover, a gradual shift has already begun. At first, the changes are subtle, almost imperceptible. A very gradual decline in muscle fibers, a slight reduction in the efficiency of muscle repair and growth. None of this is dramatic, not at first. But across decades, these small changes accumulate. And the consequences of this accumulation become increasingly meaningful over time. As muscle mass declines, so does strength. And this isn't just about lifting heavier objects, or athletic performance. Muscle strength plays a crucial role in something far more fundamental: balance and stability. As muscles weaken, the risk of falling increases. And falls, particularly in older adults, can have significant consequences, not just in the moment, but in terms of long-term mobility, independence, and overall well-being. It's a quiet reminder of just how interconnected our bodies are. A process that begins almost invisibly in our 30s, gradually shaping the kind of stability and resilience we experience decades later. Now, alongside this slow change in muscle, another quiet transformation is taking place, deep within our skeleton. Bone density decreases with age. Bones often seem like the most solid, unchanging parts of our bodies. Rigid, permanent. But in reality, bone is a living tissue, constantly being broken down and rebuilt in an ongoing cycle of renewal. In youth, this cycle tends to favor building. Bone density increases, reaching its peak relatively early in adulthood. But as the years pass, the balance gradually shifts. The process of breaking down bone tissue begins to slightly outpace the process of rebuilding it. And so, slowly, almost imperceptibly, bone density begins to decline. This doesn't happen suddenly, and for most people, it doesn't cause noticeable symptoms for a very long time. But over decades, this gradual loss of density can make bones somewhat more fragile, slightly more susceptible to fractures, particularly in certain areas of the body. It's another example of that same underlying theme, a process that unfolds slowly, quietly, in the background of everyday life, until, eventually, its effects become more apparent. And interestingly, this gradual change in bone, combined with changes elsewhere in the body, can lead to something rather unexpected. Something that many people don't realize happens at all. We tend to think of height as something fixed. Something that's determined in childhood and adolescence, and then simply stays the same for the rest of our lives. But that's not quite the full picture. As we age, many people experience a gradual decrease in height. And one of the main reasons for this lies in the spine. Between each of the vertebrae in our spine, there are small, cushion-like structures called discs. These discs are made up of a soft, somewhat gel-like core, surrounded by a tougher outer layer. They act as shock absorbers, allowing the spine to move, bend, and flex, while also helping to support the weight of the body above. Over time, these discs can undergo a kind of gradual compression. They may lose some of their hydration, becoming slightly thinner and less cushioned than they once were. And because there are so many of these discs stacked along the length of the spine, even small amounts of compression in each individual disc can add up to something noticeable when measured across the entire spine. This, combined with subtle changes in posture and bone structure over time, contributes to a gradual loss of height as we age. And the numbers, when you look at them, are rather striking. Many people lose around 2.5 centimeters, roughly 1 inch, by the time they reach older age. It's a small number, in some ways. A single inch, spread out across decades of life. But it's also a wonderfully tangible reminder of just how much quiet change happens within our bodies over time, change that often goes completely unnoticed, until one day, almost by accident, we realize that we're standing just slightly shorter than we once were. And in many ways, this is the perfect place for us to pause and reflect. Because everything we've explored so far, senescent cells releasing their quiet signals, muscle slowly beginning its decline in our 30s, bones gradually shifting their balance of renewal, and even the slow compression of the very discs that support our spine, none of these processes happen in isolation. They're all part of the same, deeply interconnected story. A story written not in dramatic moments, but in countless small, quiet changes, unfolding, year after year, within the remarkable, ever-changing structure of the human body. And as we continue this journey, we'll explore even more of these changes. How they ripple outward, how they connect to one another, and what they reveal about the long, slow, endlessly intricate process of growing older. There's a certain quiet wisdom in the way our bodies move. The way a hand reaches for a cup. The way a knee bends to climb a step. The way shoulders rotate, hips pivot, fingers curl and release. So much of this movement happens without a single conscious thought, a kind of effortless choreography, performed thousands of times a day, for an entire lifetime. And much of what makes this possible happens at the joints. A joint is, in many ways, a small marvel of engineering. It's the place where two bones meet, and yet, instead of grinding against one another, they glide. Smoothly. Quietly. And much of this smoothness comes down to a remarkable tissue called cartilage. Cartilage covers the ends of bones within a joint, acting almost like a cushion, a smooth, slightly spongy surface that allows bones to move against each other with very little friction. In youth, this cartilage tends to be thick, resilient, and well hydrated. But as the years pass, cartilage begins to thin. This thinning happens slowly, gradually, cell by cell, layer by layer, over the course of decades. And as cartilage becomes thinner, the cushioning it provides becomes a little less generous. Bones that once moved with effortless smoothness may begin to move with just slightly more resistance. This is part of why joints often become stiffer with age. And cartilage isn't the only factor at play. Within each joint, there's also a thin layer of fluid, a kind of natural lubricant, known as synovial fluid. This fluid helps reduce friction even further, allowing the surfaces within the joint to glide smoothly against one another. But just like so many other systems we've explored, the quality of this fluid can change over time. As we age, synovial fluid may become somewhat less abundant, or slightly less effective at providing that smooth, frictionless movement it once offered so easily. Combined with the gradual thinning of cartilage, this can contribute to a noticeable shift in how joints feel and how they move. And the cumulative effect of these changes is a gradual decrease in range of motion. This doesn't happen suddenly. It's not as though one day, a joint simply stops moving the way it used to. Instead, it's a slow narrowing, a gentle tightening, almost. Movements that once felt completely effortless may begin to require just a little more care. A little more warming up. A little more attention. It's another quiet example of that same underlying pattern we've seen again and again throughout this journey. Small changes, at the cellular level, gradually shaping the way we move through the world, decades later. Now, let's shift our attention to something that beats, quietly and tirelessly, for an entire lifetime. The heart. And the vast network of vessels that carry blood throughout the body. If you could observe a young, healthy artery up close, you might be surprised by how flexible it is. Arteries aren't rigid tubes. They're elastic structures, designed to expand slightly with each heartbeat, and then relax again, almost like a gentle, rhythmic breathing. This elasticity serves an important purpose. It helps absorb the force of each heartbeat, smoothing out the pressure of blood as it moves through the body. But as we age, arteries gradually become stiffer. The walls of the arteries, once so flexible and responsive, begin to lose some of that elasticity. They become slightly less able to expand and contract with each beat of the heart. And this change, though it happens slowly, over many years, has consequences that ripple outward through the entire cardiovascular system. Because when arteries become stiffer, the heart has to work harder. Think of it this way. If a tube is flexible, it can absorb some of the pressure generated by each heartbeat, distributing that force more gently. But if the tube becomes stiffer, less able to expand, that pressure has fewer places to go. And so, the heart must generate more force, with each beat, to keep blood moving through the body in the same way it always has. Over time, this added effort takes its toll. And alongside this, something else begins to shift as well, a gradual decline in what's known as cardiorespiratory efficiency. Cardiorespiratory efficiency refers to how effectively the heart and lungs work together to take in oxygen and deliver it throughout the body. In youth, this system tends to operate with remarkable efficiency. Oxygen is absorbed quickly, transported efficiently, and used effectively by tissues throughout the body. But with age, this efficiency gradually declines. The lungs may become slightly less elastic. The heart may pump slightly less blood with each beat. And the muscles themselves may become slightly less efficient at using the oxygen that's delivered to them. None of these changes are dramatic on their own. But together, they contribute to something many people notice as they grow older. A sense that physical exertion requires just a little more effort than it used to. That recovery, after exercise, takes just a little longer. It's the body's cardiovascular and respiratory systems, gently, gradually, recalibrating as the decades go by. And closely connected to all of this is something else. Blood pressure. As arteries become stiffer, and as the heart works harder to maintain circulation, blood pressure tends to rise. This is, in many ways, a direct consequence of everything we've just explored. Stiffer arteries mean less flexibility to absorb the force of each heartbeat. A harder working heart means more force being generated with each pump. And together, these changes can gradually push blood pressure upward, over time. For many people, this rise in blood pressure happens so gradually that it's almost unnoticeable from one year to the next. It's only when looking back across decades that the full extent of this gradual shift becomes clear. Now, let's turn our attention to another system entirely, one that operates quietly, in the background, every single day, protecting us from threats we're often not even aware of. The immune system. In youth, the immune system tends to operate with remarkable precision. It identifies threats, viruses, bacteria, and other potential dangers, and responds quickly and effectively to neutralize them. But as we age, the immune system gradually weakens. Scientists have a specific term for this: immunosenescence. This word combines two ideas we've already encountered. Immune, referring to the body's defense system, and senescence, that same concept of cellular aging we explored earlier. Immunosenescence describes a gradual decline in the immune system's ability to respond effectively to new threats. The production of certain immune cells may decrease. The ability to recognize and respond to unfamiliar pathogens may become less precise. And the overall responsiveness of the immune system may become somewhat slower and less robust than it once was. This doesn't mean the immune system stops working. Not at all. But it does mean that, over time, it becomes somewhat less effective at performing the tasks it once carried out with such precision. And interestingly, alongside this gradual weakening, something else tends to increase. Something that, at first, might seem contradictory. Even as the immune system becomes less effective at responding to new threats, overall levels of inflammation in the body tend to rise with age. Researchers have given this phenomenon a rather memorable name, inflammaging. A blend of inflammation and aging. Inflammaging refers to a kind of chronic, low-grade inflammation, a persistent, background level of inflammatory activity that tends to increase as we grow older. Unlike the acute inflammation we experience after an injury or during an infection, inflammaging is more subtle, more persistent. A quiet hum, rather than a sudden alarm. And remember those senescent cells we explored earlier? The ones that remain active, releasing inflammatory signals into their surroundings? They play a meaningful role here too. As senescent cells accumulate over time, the signals they release contribute to this broader pattern of chronic, low-level inflammation throughout the body. It's another beautiful example of just how interconnected these processes truly are. Changes at the cellular level, rippling outward, shaping the function of the immune system, which in turn influences inflammation throughout the body, which in turn influences nearly every other system we've explored. And finally, let's turn our attention to the most complex structure in the known universe: the human brain. As we age, certain regions of the brain undergo a gradual process of shrinkage. This might sound concerning at first, but it's important to understand this process within its proper context. Brain shrinkage, with age, is a normal and well-documented part of the aging process, not necessarily a sign of disease, but simply another example of the kind of gradual change we've explored throughout this journey. Certain regions of the brain, particularly those involved in memory, and certain aspects of cognitive processing, may show more noticeable changes in volume over time compared to other regions. This shrinkage happens slowly, gradually, over the course of many years, and even decades. And it connects to many of the processes we've already explored: cellular aging, declining efficiency in repair systems, the gradual accumulation of senescent cells, and the chronic, low-level inflammation we just discussed. It's a quiet reminder that the brain, despite often feeling like such a separate, almost mysterious part of ourselves, is deeply connected to everything else we've explored in this journey. The same underlying processes that shape our muscles, our bones, our joints, our arteries, and our immune system shape our brains as well. And so, As we draw this chapter to a close, let's take a moment to appreciate just how much we've covered. We've journeyed from stiffening joints to the quiet thinning of cartilage, from the gradual stiffening of arteries to a heart that works just a little harder year after year. We've explored the slow decline of cardiorespiratory efficiency, the gentle rise of blood pressure, and the gradual recalibration of an immune system that has spent a lifetime protecting us. We've encountered inflammaging, that quiet, persistent hum of inflammation that grows louder with time. And we've arrived, finally, at the brain itself, witnessing how even this most remarkable of organs is shaped by the same slow, intricate processes we've followed throughout this entire journey. None of these changes happen in isolation. Each one connects to the others, threads in the same vast, intricate tapestry we've been exploring from the very beginning. And as we continue forward, we'll keep following these threads. Deeper into the body. Further into the science. Uncovering, piece by piece, the remarkable, endlessly fascinating story of what it truly means to grow older. Inside the brain, there exists a network unlike anything else in the known universe, billions of neurons, connected by trillions of synapses, constantly exchanging signals in a vast, intricate symphony of communication. Every thought, every memory, every movement of a finger or flicker of an eye, begins here, in this endless conversation between cells. And as we age, this conversation begins to change. Neural communication becomes less efficient. It's a subtle shift, at first. The connections between neurons, known as synapses, may become slightly less responsive over time. The insulation around certain nerve fibers, a substance called myelin, may gradually thin in places, allowing electrical signals to travel just slightly less efficiently than they once did. None of this happens suddenly. It's not as though the lights flicker, or the signal drops out. It's more like a very slight delay. A whisper that takes a fraction of a second longer to be heard. And this connects directly to something many people notice as they grow older, a gradual change in processing speed. Processing speed refers to how quickly the brain can take in information, make sense of it, and respond. In youth, this tends to happen almost instantaneously, a kind of effortless fluency. But with age, this speed tends to gradually decline. This doesn't mean the brain becomes less capable, in any overall sense. Many cognitive abilities, knowledge, vocabulary, accumulated experience, often remain stable, or even continue to grow, well into older age. But the sheer speed at which the brain processes new information tends to slow, just slightly, decade by decade. It's a little like the difference between a familiar road you've driven 1,000 times and a brand new route you're seeing for the first time. The familiar road, you can navigate almost without thinking. The new route takes just a little more time, a little more attention. As we age, that new route feeling becomes slightly more common in everyday cognitive tasks. Now, let's turn our attention to something that touches every single one of us, every single night: sleep. Sleep is often described as one of the most restorative processes the body undergoes. And throughout life, the quality and timing of sleep can shift in some rather interesting ways. One of the most noticeable changes is that sleep cycles tend to shift earlier with age. Many older adults find themselves naturally feeling tired earlier in the evening and waking up earlier in the morning, often without an alarm. This isn't simply a matter of habit or choice. It reflects a genuine shift in the body's internal timing systems. And this connects to something called the circadian rhythm. The circadian rhythm is, in essence, the body's internal clock, a roughly 24-hour cycle that governs not just sleep and wakefulness, but also things like body temperature, hormone release, and even digestion. This rhythm is influenced by external cues, particularly light and darkness, but it's also deeply rooted within our own biology. As we age, circadian rhythms tend to weaken. The signals that once created such a clear, strong distinction between day and night within the body may become somewhat less pronounced. The internal clock may become slightly less precise, slightly less responsive to external cues like sunlight. And this can contribute to some of the changes in sleep patterns we just explored: earlier bedtimes, earlier wake times, and sometimes a sleep pattern that feels a little lighter or more fragmented than it once did. It's yet another example of that same gentle theme: systems that once operated with strong, clear rhythms, gradually becoming a little softer, a little less defined, over time. Now, let's turn to something deeply personal, something connected to memory, to comfort, to some of our most vivid experiences. The senses of taste and smell. Few things connect us to memory quite like scent. The smell of rain on warm pavement. The aroma of a particular dish from childhood. These sensory experiences are powerful, and they're made possible by an extraordinarily sensitive system of receptors within the nose and on the tongue. But as we age, taste and smell tend to gradually decline. The number of receptor cells responsible for detecting different tastes and smells may decrease slightly over time. And the way signals from these receptors are processed by the brain may also become somewhat less sensitive. This doesn't mean these senses disappear entirely. But flavors that once seemed vivid and distinct may become slightly more muted. Scents that once stood out clearly may become a little harder to detect. It's a quiet shift, one that often goes unnoticed for a long time, simply because it happens so gradually, meal by meal, year by year. And alongside taste and smell, another sense often changes as well. One that plays a profound role in how we connect with the world, and with each other. Hearing. Hearing loss becomes increasingly common with age. Within the inner ear, there are incredibly delicate structures, tiny hair cells, responsible for detecting sound waves and converting them into signals the brain can interpret. These hair cells are remarkable, but they're also somewhat delicate. And over a lifetime of exposure to sound, they can gradually become less responsive. This often happens first with higher-pitched sounds, certain consonants in speech, for example, or higher musical notes, before gradually extending to a broader range of frequencies. For many people, this happens so slowly that it's barely noticeable day to day. It's often only in certain situations, a crowded room, a quiet conversation, a particular pitch of voice, that the change becomes more apparent. And just as hearing can change with age, so too can vision. Vision can decline due to a combination of factors, including changes in blood vessels and changes within the cells of the eye itself. The eye relies on an incredibly intricate network of tiny blood vessels to deliver oxygen and nutrients to its various structures. As we age, these blood vessels, like blood vessels elsewhere in the body, may undergo some of the same changes we explored earlier. Slight stiffening, slightly reduced efficiency. At the same time, the cells within the eye itself, particularly within structures like the lens and the retina, may undergo their own gradual changes related to many of the same cellular processes we've explored throughout this journey. Declining repair efficiency. Oxidative stress. The slow accumulation of cellular damage over time. Together, these factors can contribute to gradual changes in vision, changes that, for many people, become more noticeable as the decades pass. Now, let's shift our attention to somewhere rather unexpected. Somewhere that might not seem, at first, connected to aging at all. The gut. Within the digestive system, there exists an extraordinarily complex community of microorganisms, bacteria, and other microbes, collectively known as the gut microbiome. This community plays a remarkable role in overall health. It helps with digestion. It interacts with the immune system. It even produces certain compounds that can influence processes elsewhere in the body. And as we age, the composition of this microbial community tends to change. This shift is sometimes referred to as dysbiosis, a term that describes an imbalance or alteration in the typical composition of the gut microbiome. The relative abundance of different microbial species may shift. Some species that were once more dominant may become less so. And new patterns may emerge within this vast, invisible ecosystem that exists within each of us. These changes don't happen in isolation either. They connect to many of the processes we've already explored: changes in immune function, changes in inflammation, and changes in how the body processes nutrients. So, when we step back and look at everything we've explored across this entire chapter, a clear and rather profound pattern begins to emerge. Each of these changes, slower neural communication, shifting sleep patterns, weakening circadian rhythms, declining taste and smell, hearing loss, changes in vision, and shifts within the gut microbiome, each of these, on their own, might seem like a relatively small, isolated change. But together, they contribute to something larger. These changes increase vulnerability to disease. It's an important distinction to understand. Aging itself isn't a disease, but the cumulative effect of all these gradual changes creates an environment within the body where certain diseases become more likely to emerge and to progress. And this leads us to one final, important observation, one that ties together everything we've explored, not just in this chapter, but throughout this entire journey. As people age, multiple chronic conditions often begin to appear together, rather than in isolation. A person might experience changes in cardiovascular health, alongside changes in metabolism, alongside changes in joint function, alongside changes in cognitive processing, not as separate, unrelated events, but as interconnected expressions of the same underlying processes. Researchers sometimes refer to this clustering of conditions as multimorbidity, and it reflects something we've returned to again and again throughout this entire exploration. Aging isn't a single process, happening in a single place, at a single moment. It's a vast, interconnected web, cellular changes, rippling outward into tissues, tissues, rippling outward into organs and systems, and these systems, in turn, connecting with one another, in ways that shape the entire experience of growing older. From the slowing of neural signals, to the shifting rhythms of sleep, from the quiet fading of taste, and smell, and sound, and sight, to the invisible ecosystems within our gut, every thread we've followed leads back to the same central truth. The human body is not a collection of separate parts, aging independently. It's a single, deeply interconnected whole. Every system, every cell, every quiet biological process, woven together into one long, continuous, endlessly fascinating story. And as we continue this journey, we'll keep following that story. Deeper still. Into new chapters, new systems, and new layers of what it truly means to grow older. Throughout this journey, we've explored the body almost like a vast, interconnected machine, cells, tissues, organs, all moving through time according to certain shared patterns. And it would be easy, at this point, to imagine aging as something fixed. Something written, long ago, into the code of our DNA, a kind of biological destiny, set in motion at birth, and simply unfolding exactly as planned. But here's where the story takes a rather surprising turn. Because when researchers actually studied this question, when they looked closely at why some people seem to age more quickly, and others more slowly, they found something that challenges this assumption quite significantly. Genetics, it turns out, explains only around 10 to 15% of the variation in how people age. Let that number settle for a moment. 10 to 15%. For something so often described as being written in our genes, aging turns out to be remarkably less genetically determined than many people assume. And this pattern holds true even when researchers look specifically at longevity itself, how long people tend to live. Studies suggest that longevity is also roughly 15% heritable, which means the vast majority of what shapes how we age and how long we live comes from somewhere else entirely. So, where does it come from? The answer, according to a growing body of research, points towards something we have far more influence over than we might expect: lifestyle. Lifestyle appears to be the dominant influence on the speed at which we age. Now, lifestyle is a broad word, and it's worth pausing to appreciate just how much it encompasses. It includes the food we eat, certainly. The amount and quality of sleep we get. How physically active we are throughout our days. Whether we smoke or drink, and to what degree. But it also includes things that are a little less obvious: patterns of stress, the quality of our relationships, the environments we spend our time in, and even the way we think about our own lives. All of these threads, woven together, day after day, year after year, appear to play a far larger role in shaping our biological age than the genetic code we were born with. And this connects to something rather beautiful when you think about it. Aging isn't simply something that happens to us, passively, like a sentence being carried out. It's something that's shaped, continuously, by the life we actually live. Aging is shaped by lifelong environment and behavior. Not a single decision, made once. Not a single year of particularly healthy or unhealthy living. But the accumulated pattern of countless small choices and countless environmental influences, repeated again and again across an entire lifetime. It's a little like the slow shaping of a riverbed by water flowing over it for centuries. No single drop of water carves the canyon. But each drop, repeated endlessly, over time, gradually shapes something profound. Now, this idea, that environment and behavior shape aging, leads us to something perhaps even more unexpected. Because it's not just about individual choices, made in isolation. It's also about the broader context in which those choices take place. Researchers have found that socioeconomic factors influence biological aging rates. This is a significant finding, and it's worth sitting with for a moment. It suggests that the conditions a person lives within, their access to resources, their education, their economic stability, the environments they spend their time in, can leave a measurable imprint on the biological processes of aging itself. And more specifically, research has shown that disadvantaged environments can accelerate epigenetic aging. Remember those epigenetic clocks we explored earlier? Those remarkable tools, capable of measuring biological age, based on patterns within our DNA. It turns out, the environments people live within, particularly environments shaped by economic disadvantage or chronic stress, can influence these very patterns. People living in more disadvantaged circumstances may show signs of faster epigenetic aging, their biological clocks, in a sense, ticking just slightly faster than their chronological age would suggest. It's a profound finding, one that extends the story of aging beyond the individual and into the broader fabric of society itself. But within this larger picture, there's also something deeply personal. Something that touches each of our individual lives in very direct ways. And it has to do with connection. Research has consistently shown that strong social ties reduce mortality risk. People with close relationships, friends, family, communities they feel genuinely connected to, tend to live longer, on average, than people who lack these connections. And the flip side of this finding is equally striking. Loneliness increases disease risk. This isn't simply about emotional well-being in the abstract sense, though that matters too, of course. Loneliness appears to have measurable effects on physical health itself, influencing processes like inflammation, immune function, and even cardiovascular health. It's a rather profound thought when you consider it. The quality of our relationships, the sense of connection we feel to the people around us, appears to ripple outward into the very biological processes we've been exploring throughout this entire journey. And this leads us to one final thread, one that ties together so much of what we've explored in a rather unexpected way. Research has found that psychological resilience slows biological aging. Resilience, here refers to something rather specific, the capacity to adapt, to cope, and to recover in the face of stress and adversity. And what's remarkable is that this capacity, this internal quality that can vary so much from person to person, appears to have measurable effects on the biological markers of aging we've explored throughout this journey. People who tend to respond to stress with greater resilience may show signs of slower biological aging compared to those who experience stress in more prolonged or more intense ways. And closely related to this is something perhaps even more profound. A sense of purpose in life is linked to lower mortality. People who report a strong sense of purpose, a feeling that their life has meaning and direction, tend to live longer, on average, than those who don't. It's a remarkable finding when you think about it. Something as seemingly intangible as purpose, a feeling, a perspective, a way of relating to one's own life, appears to leave a measurable imprint on the biological processes that shape how long and how well we live. So, as we draw this chapter to a close, let's take a moment to reflect on everything we've explored. We began with a number, that 10 to 15% figure, a reminder that genetics, despite playing some role in how we age, is far from the dominant force many people assume it to be. And from there, we followed a thread outward, into lifestyle, into environment, into the broader social and economic contexts that shape our daily lives, and finally, into something deeply personal. The quality of our relationships. Our resilience in the face of life's challenges. And our sense of purpose, the quiet, often unspoken sense of why we do what we do each day. None of these things, on their own, determine exactly how someone will age. Life is far too complex, far too individual, for such simple equations. But together, they paint a picture of aging that's far more open, far more influenceable than many of us might have imagined. Aging isn't simply something written into our DNA at the moment of birth, and then carried out exactly as planned. It's something shaped continuously by the lives we live, the connections we nurture, the environments we find ourselves within, and the quiet, often invisible sense of meaning that we carry with us through our days. And as we continue this journey into the chapters ahead, we'll keep following this thread, exploring in even greater depth exactly how these influences shape the long, slow, endlessly intricate story of what it truly means to grow older. There's a quiet assumption many people carry, often without realizing it, that growing older means, primarily, a slow loss. A gradual fading. Less energy, less sharpness, less joy. And yet, when researchers actually study happiness across entire lifetimes, they find something rather different. Something that often comes as a genuine surprise. Emotional well-being often increases in later life. This pattern has a name. Researchers call it the U-shaped happiness curve. Picture, for a moment, a gentle curve, beginning relatively high in youth, dipping downward through the middle years of life, often the busiest, most demanding years, filled with career pressures, raising families, and countless competing responsibilities. And then, rising again, later in life. often reaching levels of contentment and emotional well-being that match or even exceed those early years. It's a rather beautiful finding when you sit with it. The years many people associate with decline often bring with them a kind of emotional richness, a settling, a clarity, a sense of ease that wasn't always present in earlier, more turbulent chapters of life. And this isn't the only area where aging brings unexpected gains. Let's talk, for a moment, about intelligence. When most people think about cognitive aging, they tend to focus on what's lost: processing speed, memory for new information, the kind of mental quickness we explored earlier in this journey. But intelligence isn't just one thing. Researchers often distinguish between two broad types. There's fluid intelligence, the ability to reason, solve novel problems, and process new information quickly. And then, there's crystallized intelligence, the accumulated knowledge, vocabulary, and understanding built up over an entire lifetime of experience. And here's the remarkable part. While fluid intelligence may gradually decline with age, crystallized intelligence often improves. The vocabulary someone has access to, their depth of understanding in areas they've spent years exploring, their ability to draw on decades of accumulated knowledge and apply it to new situations. All of this tends to grow rather than shrink with age. It's a lovely reframing in some ways. The brain doesn't simply decline. It changes. Certain capacities may become a little less sharp, while others, built slowly over decades, become richer, deeper, and more refined. And for some people, this pattern of preserved and even enhanced cognitive function becomes especially pronounced. Researchers have identified a remarkable group of individuals, sometimes called superagers. These are people, often in their 80s and beyond, whose cognitive function, particularly memory, remains remarkably similar to that of people decades younger. Brain scans of superagers often reveal something fascinating. Certain regions of the brain, regions that, in many people, tend to shrink with age, appear remarkably preserved in these individuals. As if, in some way, their brains have followed a slightly different timeline than the one we might typically expect. Now, superagers represent a relatively small portion of the population. But their existence raises a profound question. What allows some brains to age so differently than others? And this question leads us towards something researchers have studied quite extensively, a concept known as cognitive reserve. One of the most well-established contributors to cognitive reserve is education. Education, particularly sustained engagement in learning throughout life, appears to build a kind of buffer within the brain. A reserve of cognitive resources that can be drawn upon even as certain age-related changes begin to occur. This doesn't mean education prevents brain aging entirely, but it does appear to provide the brain with additional pathways, additional connections, that can help compensate for changes that might otherwise have a more significant impact. In a sense, a brain that has spent decades actively engaging with new ideas, new information, and new challenges seems to develop a kind of resilience, a depth that helps protect it against some of the more disruptive effects of cognitive aging, including, notably, dementia. Now, let's turn our attention to something else entirely, something physical rather than purely cognitive. But something that, remarkably, connects directly back to the brain. Exercise. We often think of exercise primarily in terms of muscles and cardiovascular health. And certainly, those benefits are real and significant. But exercise also does something rather extraordinary at the molecular level. When muscles contract during physical activity, they release a particular class of molecules called myokines. Myokines act as signaling molecules, traveling through the bloodstream and influencing other tissues throughout the body. And remarkably, many of these myokines appear to have protective effects, helping to regulate inflammation, support metabolic health, and even influence processes within the brain itself. In other words, every time muscles engage in physical activity, they're not just building strength. They're sending out a kind of biochemical message, a message that ripples outward throughout the entire body. And this connects directly to something we explored earlier in this journey, inflammaging. That persistent, low-grade inflammation that tends to increase with age. Exercise reduces inflammation. And alongside this, exercise also appears to support brain health more directly, increasing blood flow to the brain, supporting the growth of new connections between neurons, and potentially even influencing some of the same processes related to cognitive reserve that we just explored. It's a rather elegant connection when you think about it. A process that begins in the muscles, ripples outward, and ultimately reaches the brain itself. Now, let's step back for a moment and consider the bigger picture. Throughout this journey, we've explored countless individual threads, cellular processes, organ systems, lifestyle factors, social connections, and now, exercise and cognitive reserve. And when researchers study these threads together across large populations over long periods of time, a consistent pattern emerges. Healthy habits consistently extend lifespan. This might sound almost deceptively simple, but the consistency of this finding across countless studies, populations, and decades of research is genuinely remarkable. People who engage in regular physical activity, people who avoid smoking, people who maintain healthy body weight and manage stress effectively, people who prioritize sleep and social connection. These individuals, consistently, across study after study, tend to live longer. And among these various habits, one, in particular, stands out as having a particularly profound influence. Diet. Diet quality strongly influences the trajectory of aging. What we eat, day after day, year after year, provides the raw materials for nearly every process we've explored throughout this journey. The proteins that make up our cells, the fats that form cellular structures, the nutrients that support repair systems and energy production within our mitochondria. A diet rich in nutrient-dense foods fruits, vegetables, whole grains, healthy fats, and adequate protein appears to support many of the protective processes we've discussed throughout this journey. While diets high in processed foods and added sugars may contribute to some of the more harmful processes like glycation and chronic inflammation, over decades, these dietary patterns accumulate, shaping in very real ways the trajectory of how someone ages. Now, as we bring this chapter toward its close, let's zoom out even further, beyond the individual entirely. Because when researchers study aging across different populations around the world, they find something important. Aging is not uniform across populations. Different populations, living in different environments, with different lifestyles, different diets, different healthcare systems, and different social structures, show different patterns of aging. Some populations show slower rates of certain age-related changes. Others show faster rates of different changes. And these differences aren't random, they reflect the cumulative influence of all the factors we've explored throughout this journey: genetics, environment, lifestyle, and social context, woven together in countless different combinations around the world. And at the most fundamental level, this variation extends all the way down to the molecular level itself. Molecular damage patterns differ between individuals. The specific ways that DNA accumulates damage. The specific patterns of epigenetic change. The specific rates of cellular senescence and inflammation. All of these vary from person to person, shaped by the unique combination of genetics, environment, and lifestyle that each person carries throughout their own individual life. So, as we close this chapter, let's take a moment to sit with everything we've explored. We began with happiness, discovering that emotional well-being often increases in later life. We explored intelligence, discovering that certain forms of cognitive ability can deepen with age. We met the superagers, individuals whose brains seem to follow a remarkably different timeline. And we explored the protective power of education, of exercise, of myokines, of healthy habits, and of diet. And finally, we zoomed out to see the bigger picture. Aging as a process that varies profoundly from population to population and from person to person, shaped at every level by the unique, intricate, endlessly varied story of each individual life. And as we continue this journey, we'll keep exploring this story, moving ever deeper into the remarkable, intricate, endlessly fascinating science of what it truly means to grow older. Earlier in this journey, we ventured into the hidden world of cells, exploring damage, repair, and the quiet processes that shape how our bodies change over time. Now, we return to that world once more, but this time with a sharper lens. Because in recent decades, scientists have begun to organize everything we know about aging into a more structured framework, a kind of map of the underlying mechanisms that drive the aging process. This framework is often referred to as the hallmarks of aging. It's a remarkable piece of scientific synthesis. Researchers, drawing together findings from countless studies across many different fields, identified a set of core biological processes that appear again and again across nearly every tissue and every organism studied. Among these hallmarks are things we've already touched on. Cellular senescence, that strange state where cells stop dividing, yet remain active. Genomic instability, the gradual accumulation of damage within our DNA. And stem cell decline, a gradual reduction in the body's ability to generate fresh, new cells, to replace those that are lost or worn out over time. Each of these hallmarks represents a thread. And as we'll see in this chapter, these threads are woven together in ways that are both intricate and surprisingly elegant. Let's begin with one of the most well-known and visually evocative of these mechanisms. Something that lives at the very tips of our chromosomes. Telomeres. Imagine, for a moment, the protective cap at the end of a shoelace, that small plastic tip that keeps the woven threads from fraying apart. Telomeres serve a remarkably similar purpose for our DNA. They're repetitive sequences of genetic material sitting at the ends of each chromosome, protecting the more vital genetic information within from becoming damaged or tangled during the process of cell division. And here's the key detail. Every time a cell divides, its telomeres shorten, just slightly. It's a bit like that shoelace tip, slowly wearing down with repeated use. Each division, each replication of the cell's DNA, takes a small toll on these protective caps. Over many divisions, across many years, telomeres become progressively shorter. For most of a cell's life, this gradual shortening doesn't cause any noticeable problems. The telomere is still long enough to do its job. But eventually, a threshold is reached. When telomeres become critically short, they trigger cellular senescence. It's almost as though the cell has an internal sensor, monitoring the length of these protective caps. And when that length drops below a certain point, the cell receives a signal. A signal that essentially says, it's time to stop dividing. This is one of the ways that senescent cells, those cells we explored in such depth earlier in this journey, actually come into existence. Not through damage alone, but through this slow, almost rhythmic process of telomere shortening, repeating with each division until a critical limit is reached. It's a rather beautiful piece of biological design, in some ways. A built-in safeguard, preventing cells from dividing indefinitely, with DNA that has become too worn down to copy safely. But like so many safeguards we've encountered throughout this journey, this protective mechanism also contributes to the broader story of aging itself. Now, let's turn our attention back to a structure we've visited before. Those tiny, tireless powerhouses within our cells. Mitochondria. We explored, earlier in this journey, how mitochondria can become less efficient with age, producing slightly less energy, while generating slightly more of those reactive molecules known as ROS. But it's worth pausing here to appreciate just how central this process truly is. Mitochondrial dysfunction doesn't just affect energy levels in isolation. It accelerates aging more broadly. Think of mitochondria almost as a kind of communication hub within the cell. They don't just produce energy, they also send signals to other parts of the cell about the cell's overall state. When mitochondria become dysfunctional, these signals change. And these changes can ripple outward, influencing processes like inflammation, cellular stress responses, and even the likelihood of a cell entering that senescent state we just discussed. In this way, mitochondrial dysfunction acts almost like an accelerant, amplifying and speeding up many of the other aging processes we've explored throughout this journey. Now, let's shift our focus to something rather different. Not damage, this time, but regulation. Specifically, the way our cells sense and respond to the nutrients available to them. This brings us to a fascinating area of research involving what are known as nutrient sensing pathways. You can think of these pathways almost like internal sensors, constantly monitoring the availability of nutrients, energy, and growth signals within the body. And based on what they detect, these pathways help determine how cells behave, whether they prioritize growth and reproduction, or instead shift toward maintenance and repair. 3 of these pathways, in particular, have received significant attention from researchers. They go by names that might sound a little technical at first: mTOR, IGF-1, and AMPK. mTOR, in simple terms, tends to promote growth and cellular activity, particularly when nutrients are abundant. IGF-1, which stands for insulin-like growth factor 1, is closely linked to growth signaling throughout the body. And AMPK, by contrast, tends to become more active when energy levels are lower, helping to shift cells toward more conservative, maintenance-focused behaviors. Together, these 3 pathways form a kind of intricate balancing system, constantly adjusting based on the nutritional and energetic state of the body. And remarkably, research has found that these pathways play a significant role in regulating the speed of aging itself. One of the most striking findings in this area involves IGF-1. Across numerous studies, in various animal species, researchers have found that reduced IGF-1 signaling is associated with extended lifespan. This finding has been observed again and again across different organisms, from tiny worms to mice. Animals with reduced IGF-1 signaling, animals whose growth signal, in a sense, has been slightly dialed down, tend to live longer than their counterparts with typical IGF-1 levels. It's a finding that, at first, might seem almost counterintuitive. We often associate growth with health and vitality. And yet, in the context of lifespan, a slightly more conservative approach to growth signaling appears to be associated with greater longevity. It's a reminder of just how complex and sometimes counterintuitive the biology of aging can be. Now, let's introduce another set of players in this story, ones with a rather elegant name: sirtuins. Sirtuins are a family of proteins that play a role in regulating several important cellular processes. Among their many functions, sirtuins help regulate DNA repair and metabolism. You might think of sirtuins almost like cellular coordinators, helping to manage how cells respond to various forms of stress, including the kind of DNA damage we discussed earlier in this journey. They help orchestrate repair processes, and they also play a role in how cells manage their metabolic activity. But here's where things become particularly interesting. Sirtuins require a particular molecule in order to function properly. A molecule called NAD+. NAD+ stands for nicotinamide adenine dinucleotide, though the full name matters far less than what it actually does. NAD+ plays a crucial role in numerous cellular processes, including energy production and the activity of sirtuins themselves. And here's the key detail for our story. NAD+ levels decline with age. As we get older, the amount of NAD+ available within our cells tends to gradually decrease. And because sirtuins depend on NAD+ to function properly, this decline in NAD+ levels can also affect how effectively sirtuins are able to carry out their roles, including their role in DNA repair and metabolic regulation. It's another example of that same interconnected pattern we've seen throughout this journey. A decline in one molecule rippling outward, affecting the function of other systems that depend upon it. Now, let's turn to something that might sound rather unexpected, something that connects the story of aging to a much older story, the story of viruses and their relationship with our own DNA. Deep within the human genome, woven into the very fabric of our DNA, exist remnants of ancient viruses. Sequences known as endogenous retroviruses. These sequences are believed to be remnants of viral infections that occurred far back in our evolutionary history, infections so ancient that their genetic material became permanently incorporated into the genomes of our ancestors and passed down generation after generation ever since. For most of our lives, these ancient viral sequences remain largely silent, inactive, a kind of dormant genetic archive tucked away within our DNA. But research suggests that during aging, these endogenous retroviruses may become reactivated. This is a relatively newer area of research, and scientists are still working to fully understand what this reactivation means for the aging process. But it's thought that as some of the regulatory systems we discussed earlier, particularly those related to chromatin structure and epigenetic regulation, become less precise with age, some of these ancient, dormant sequences may become slightly more active than they once were. And this activation may contribute to some of the inflammatory processes we discussed earlier in this journey, adding yet another thread to the complex tapestry of aging. It's a rather extraordinary thought, isn't it, that within each of us lies a kind of genetic echo of ancient viral history, and that this echo may grow slightly louder as the years pass. Now, let's turn to our final topic in this chapter, one that brings us back to something more familiar: tissue repair. Throughout this journey, we've explored many different repair systems: DNA repair, protein quality control, cellular cleanup through autophagy. But there's another form of repair worth exploring, the kind that happens when tissues are injured. When tissue is damaged, whether through injury, illness, or simply the wear of everyday life, the body initiates a repair process. In youth, this process often results in tissue that's restored relatively close to its original structure and function. But with age, this repair process can change. Specifically, fibrotic scarring can impair tissue repair. Fibrosis refers to the formation of excess connective tissue, a kind of scar tissue that forms in place of the original, more specialized tissue that was there before. In youth, some degree of scarring is a normal part of healing. But as we age, this process can become somewhat exaggerated. More connective tissue may form than is strictly necessary. And this excess tissue, rather than restoring the original function of the damaged area, can actually impair it. Think of it almost like patching a hole in a piece of fabric, but using a patch that's stiffer and less flexible than the original material. The hole is covered, but the fabric as a whole no longer moves quite the way it used to. Across many tissues, the heart, the lungs, the liver, and others, this gradual shift toward more fibrotic, less functional repair can contribute to declining function over time. And so, as we draw this chapter to a close, let's take a moment to step back and look at everything we've explored. We began with telomeres, those small, protective caps, gradually shortening with each cell division, eventually triggering the senescence we explored so deeply earlier in this journey. We revisited mitochondria, seeing how their dysfunction doesn't just affect energy levels, but accelerates the broader aging process. We explored the intricate nutrient-sensing pathways, mTOR, IGF-1, and AMPK, discovering the rather surprising relationship between reduced growth signaling and extended lifespan. We met the sirtuins and their dependence on NAD+, a molecule that quietly declines with age. We encountered the ancient echoes of endogenous retroviruses, dormant for most of our lives, yet potentially reawakening as the years pass. And finally, we explored fibrosis, the way our body's repair processes themselves can change over time, sometimes in ways that create more problems than they solve. Each of these mechanisms represents a piece of an extraordinarily complex puzzle. And yet, when viewed together, they begin to form something remarkably coherent. A picture of aging not as a single process, but as a deeply interconnected system of mechanisms influencing one another in ways that researchers are only beginning to fully understand. And as we continue this journey, we'll keep exploring this system, moving ever deeper into the remarkable, intricate, endlessly evolving science of what it truly means to grow older. When tissue is injured, healing begins almost immediately. Cells migrate to the site of damage, new structures form, and gradually, function is restored. It's a process most of us take entirely for granted, especially when we're young, because it tends to happen so reliably, so quickly, that we barely notice it at all. But as we age, tissue repair becomes less efficient. This builds directly on what we explored in the last chapter. With fibrosis tending to increase, with stem cells becoming less abundant and less responsive, and with cellular communication growing less precise, the entire repair process begins to slow down. Wounds that might have healed in days during youth may take noticeably longer in later life. And the quality of that repair, the degree to which tissue returns to its original strength and flexibility, often declines as well. This isn't a failure of any single system. It's the cumulative result of many systems, each working just a little less smoothly than before, all contributing to the same outcome. And this brings us to something worth sitting with for a moment, because it's really the thread that ties so much of this journey together. Aging involves a breakdown in systemic communication. Throughout this entire exploration, we've talked about individual cells, individual tissues, individual organs. But none of these exist in isolation. They're constantly talking to one another, through hormones, through signaling molecules, through the kind of myokines we discussed when we explored exercise, through countless chemical messages traveling through the bloodstream every second of every day. In youth, this communication tends to be remarkably coordinated. Organs respond to each other's signals quickly and appropriately. But with age, this coordination begins to fray, just slightly. Signals may become less precise. Responses may become slower, or less proportionate. It's a bit like a large orchestra, where most of the musicians are still playing beautifully, but the timing between sections has become just a touch looser than it once was. And this loss of coordination helps explain something we touched on earlier: why so many age-related changes seem to cluster together, rather than appearing as isolated events. When the communication network itself becomes less precise, its effects ripple outward, touching many systems at once. Now, let's turn to a system that ages in a rather distinctive way, one that follows its own particular timeline. The ovaries. Ovarian aging follows unique molecular pathways. Unlike many other tissues in the body, which tend to decline gradually, and relatively continuously, over many decades, the ovaries follow a somewhat different pattern. They contain a finite number of egg cells, established very early in life, and this number naturally declines over time, through processes that are, in some ways, distinct from the aging mechanisms we've explored in other tissues. Researchers have found that ovarian tissue can show signs of accelerated aging, relative to other tissues in the same body, at the same chronological age. The molecular signals involved, the rate of cellular changes, the way senescence unfolds within this tissue specifically, all of this appears to follow its own particular rhythm. It's a fascinating reminder that aging isn't a single, uniform clock ticking at the same pace throughout the body. Different tissues can age along somewhat different trajectories, shaped by their own unique biology and their own unique demands. Now, let's return to something we touched on earlier in this journey, but look at it from a slightly different angle. Blood vessels, and the role they play in delivering what tissues need to survive. Vascular aging reduces tissue perfusion. Perfusion is simply the term for how well blood flows through a particular tissue, delivering oxygen and nutrients while carrying away waste products. And as blood vessels age, becoming stiffer and somewhat less responsive, the efficiency of this delivery system can decline. Even small reductions in perfusion can have meaningful effects, particularly for tissues with high energy demands. The brain, the heart, the muscles, all of these depend on a steady, well-regulated supply of blood. When that supply becomes slightly less efficient, even subtly, it can influence how well these tissues are able to maintain themselves, repair damage, and function under stress. It's a quiet kind of change, often unnoticed for years. But over time, it becomes another thread woven into the broader pattern we've followed throughout this journey. And this leads us to something that, statistically, has an enormous influence on human health and longevity. Cardiovascular aging drives many diseases. We explored, earlier, how arteries stiffen with age, how the heart works harder, and how blood pressure tends to rise. These changes don't simply stay confined to the cardiovascular system itself. Because the cardiovascular system touches every other organ, supplying blood, oxygen, and nutrients throughout the body, changes here can have outsized effects elsewhere. Many of the conditions that become more common with age, conditions affecting the brain, the kidneys, the eyes, and other organs, are closely linked to changes in the cardiovascular system. In many ways, the health of our blood vessels and heart serves as a kind of foundation, upon which the health of many other systems depends. This is part of why cardiovascular health has received so much attention in research focused on aging and longevity. It's not just one system among many. It's a system whose influence extends, quite literally, everywhere blood travels, which is to say, everywhere in the body. Now, before we move into the final section of this chapter, let's pause on something that connects everything we've discussed so far to the future of this field. Biomarkers of aging are improving rapidly. Throughout this journey, we've mentioned various ways researchers measure biological age. Epigenetic clocks, markers of inflammation, measures of mitochondrial function. And in recent years, the number and precision of these biomarkers has been expanding remarkably quickly. This matters more than it might first appear. Because in order to understand whether something, a lifestyle change, a medication, an intervention of any kind, actually slows aging, researchers need reliable ways to measure aging itself, ideally without waiting decades to observe outcomes like disease or mortality directly. As these biomarkers become more refined, researchers are increasingly able to detect subtle shifts in biological age, sometimes over the course of months, rather than years. And this capability is opening the door to something that, until quite recently, would have seemed almost like science fiction. The ability to actually test and measure interventions designed to influence the aging process itself. Which brings us, rather naturally, to where we're headed next. For most of human history, aging was viewed as something fixed, inevitable. A process that happened to every living thing, in much the same way, regardless of circumstance. But research over the past few decades has begun to challenge that view, quite profoundly. aging is now considered to be biologically plastic. The word plastic, here, doesn't refer to the material. It refers to the idea of something being changeable, moldable, responsive to influence, rather than fixed and unchangeable. And this represents a genuinely significant shift in how scientists think about aging. Rather than viewing it as a single, predetermined timeline, researchers increasingly view aging as a process that can be influenced, adjusted, and in some cases, meaningfully altered. This idea connects directly to something we explored earlier, when we discussed how lifestyle, environment, and social factors appear to play such a dominant role in shaping how people age. If aging were entirely fixed, these factors wouldn't matter nearly as much as research suggests they do. And this leads to a finding that has shaped much of modern aging research. The rate of aging can be modified experimentally. In laboratory studies, across many different organisms, researchers have found that specific interventions, genetic changes, dietary modifications, certain compounds, can measurably alter how quickly an organism ages. In some cases, these interventions extend lifespan significantly. In others, they improve what researchers call health span, the period of life spent in good health, free from major disease or disability. This doesn't mean aging can simply be switched off. But it does mean that the rate at which aging unfolds, the speed of that gradual accumulation of changes we've followed throughout this entire journey, isn't fixed in stone. It can shift, depending on the conditions an organism experiences. And one of the most well-studied of these interventions is something that, on the surface, sounds almost deceptively simple. Caloric restriction extends lifespan in many species. Caloric restriction refers to consuming fewer calories than would typically be eaten, while still maintaining adequate nutrition, avoiding malnutrition. And across an extraordinary range of species, from simple organisms like yeast, to worms, to flies, to mice, and even, to some extent, in primates, caloric restriction has been shown to extend lifespan, sometimes quite significantly. The mechanisms behind this effect connect to many of the pathways we explored in the previous chapter. Caloric restriction appears to influence nutrient-sensing pathways, like mTOR and AMPK, shifting cells toward a more conservative, maintenance-focused state. It appears to support processes like autophagy, that cellular cleanup system we discussed earlier. And it appears to influence inflammation and metabolic health in ways that, collectively, seem to slow several aspects of the aging process. Now, sustained caloric restriction, as a long-term lifestyle, is genuinely difficult for most people to maintain. But this research has opened the door to exploring related approaches, ones that might capture some of the same benefits, in ways that are more practical for everyday life. And this brings us to our final point for this chapter. Intermittent fasting activates autophagy. Rather than continuously restricting calories, intermittent fasting involves cycling between periods of eating and periods of not eating, often within a structured daily or weekly pattern. During these fasting periods, particularly once a certain duration has passed, the body's nutrient-sensing pathways shift. Levels of certain hormones change. And notably, autophagy, that cellular cleanup process we explored earlier tends to become more active. It's a rather elegant idea when you think about it. During periods without incoming nutrients, cells appear to shift their priorities away from growth and toward maintenance, cleanup, and repair. As though, in the absence of new material to work with, cells turn their attention inward, tidying up, clearing away what's no longer needed. Research into intermittent fasting is still evolving, and scientists are continuing to explore exactly how these patterns translate into long-term effects on human aging and health. But the underlying connection between periods of fasting and increased cellular cleanup represents one of the more promising threads in this rapidly developing field. And so, as we draw this chapter to a close, we find ourselves standing at a rather remarkable threshold. We've journeyed through tissue repair, gradually becoming less efficient with age. Through the loss of precise communication between organs and systems. Through the unique aging pathways of the ovaries, and the far-reaching influence of vascular and cardiovascular aging. And we've seen how the tools for measuring aging itself are advancing rapidly, opening new possibilities for understanding this process more deeply than ever before. And from there, we stepped into something new. The idea that aging, rather than being fixed, is biologically plastic. That its rate can be modified through careful scientific study. And that interventions, from caloric restriction to intermittent fasting, are beginning to reveal just how responsive this process truly is to the conditions we create within our own bodies. It's a quiet but profound shift in how we understand what it means to grow older. And in the chapters ahead, we'll continue following this thread, deeper still, into the science of interventions, possibilities, and the evolving understanding of just how much influence we may have over the long, intricate, endlessly fascinating journey of human aging. We've spent a great deal of time throughout this journey exploring exercise from various angles. Its effects on muscle, on the cardiovascular system, on inflammation, and on the brain. But it's worth pausing here to appreciate something that ties all of this together. Exercise slows multiple aging pathways. This isn't simply a collection of separate benefits, happening to coincide with one another. When researchers look closely, they find that physical activity appears to influence many of the core mechanisms we explored in earlier chapters, often simultaneously. It supports mitochondrial function. It influences those nutrient-sensing pathways, like AMPK, that we discussed when exploring mTOR and IGF-1. It reduces inflammation. It supports autophagy. In a sense, exercise touches nearly every thread we've followed throughout this entire journey, all at once. Not as a single targeted intervention, but as something that gently nudges the whole system toward a state of better maintenance and slower decline. And this idea of intervening in the underlying mechanisms of aging itself, rather than simply treating individual symptoms as they arise, brings us to one of the most actively studied areas in modern aging research. We spent considerable time, earlier in this journey, exploring senescent cells. Those cells that stop dividing, yet remain stubbornly active, releasing inflammatory signals into their surroundings, and gradually accumulating in tissues throughout the body. and researchers have developed a class of compounds designed specifically to address this. They're called senolytics. Senolytics remove harmful senescent cells. The basic idea is rather elegant. Senescent cells, despite remaining metabolically active, rely on certain survival pathways to avoid the kind of programmed cell death that would normally clear away damaged or unnecessary cells. Senolytic compounds work by interfering with these specific survival pathways in a way that's relatively selective for senescent cells, while leaving healthy, functioning cells largely unaffected. In laboratory studies, this approach has shown some genuinely striking results. By selectively clearing out these lingering, dysfunctional cells, researchers have observed improvements in various measures of tissue health and function. Now, alongside senolytics, researchers have also developed a related, but somewhat different approach. Xenomorphic drugs suppress inflammatory signaling. Rather than removing senescent cells altogether, xenomorphic compounds work by targeting the SASP, that senescence-associated secretory phenotype we discussed earlier. Remember, senescent cells don't just sit quietly. They release a stream of inflammatory signals into their surroundings. Xenomorphic drugs aim to quiet this signaling without necessarily eliminating the cells themselves. It's a bit like turning down the volume on a disruptive broadcast, rather than removing the broadcaster entirely. Both approaches, senolytics and xenomorphics, represent attempts to address the same underlying issue, the accumulation and activity of senescent cells, but through somewhat different mechanisms. And the results, so far, in animal studies, have been notable. These therapies improve health span in animals. In various studies, animals treated with senolytic or xenomorphic compounds have shown improvements across a range of measures, things like physical function, tissue health, and resistance to certain age-related conditions. Importantly, much of this research focuses not just on lifespan, how long animals live, but on health span, the quality of those years, and the period of time spent free from significant decline or disease. This distinction matters a great deal, because extending lifespan alone, without considering health span, wouldn't necessarily represent the kind of progress most people would hope for. What these studies suggest is something more meaningful, the possibility of extending the years of healthy, functional life, rather than simply extending life itself. And this growing interest in senescent cells connects to something rather significant when we consider the broader landscape of human health. Senescent cells contribute to many diseases. Throughout this journey, we've explored how senescent cells release inflammatory signals, contribute to chronic, low-grade inflammation, and influence the function of nearby tissues. And when researchers look across a wide range of age-related conditions affecting the heart, the joints, the lungs, the brain, and many other systems, they consistently find signatures of cellular senescence playing a role. This has led to a shift in how some researchers think about disease itself. Rather than viewing each age-related condition as a separate problem, with its own separate cause, there's growing interest in the idea that senescent cells may represent a kind of shared, underlying contributor across many different conditions. Address this shared mechanism, and you might, in principle, influence multiple conditions simultaneously. It's a rather different way of thinking about disease, one that focuses less on individual diagnoses and more on the shared biological processes that underlie so many of them. Now, let's turn to something that represents, perhaps, one of the more striking developments in this entire field. Something that touches on a question we explored very early in this journey, the idea of epigenetic clocks and biological age. Cellular reprogramming can partially reverse cellular age. This is a remarkable concept, and it's worth taking a moment to really sit with what it means. Cellular reprogramming refers to a process by which mature, specialized cells can be coaxed back toward a more youthful state. This involves activating certain factors within the cell, factors that influence how genes are expressed and how the cell's overall identity is maintained. And in various studies, researchers have found that this process of reprogramming can partially reverse markers of cellular age, things like those epigenetic clocks we discussed at the very beginning of this journey. Cells that, by these measures, appeared older have shown shifts toward patterns more typical of younger cells following reprogramming. This connects directly to something we touched on in an earlier chapter. Epigenetic changes may be reversible. For much of this journey, we've described epigenetic changes, those small chemical marks that accumulate on our DNA over time, as something that simply happens as part of aging. A kind of drift away from a more youthful pattern. But this newer research suggests something rather different. If these changes can be partially reversed through reprogramming, then perhaps the patterns themselves aren't simply a fixed, one-way drift. Perhaps, in some circumstances, they can be guided back toward an earlier state. And this leads to a finding that researchers have described in rather striking terms. Restoring epigenetic integrity can rejuvenate cells. When researchers have used reprogramming approaches to restore certain epigenetic patterns toward more youthful configurations, they've observed genuine functional improvements in those cells. Improvements in things like how efficiently cells repair damage, how effectively they respond to stress, and how well they carry out their normal functions. It's not simply that the cells look younger by certain molecular measures. In some cases, they appear to behave in ways more characteristic of younger cells as well. And this brings us to a particularly significant piece of this puzzle. One we touched on in the previous chapter when discussing chromatin. Chromatin restoration reverses signs of aging in mice. Remember, chromatin refers to the structure formed when DNA wraps around proteins within the cell's nucleus, a kind of organized packaging that influences which genes are accessible and when. In studies involving mice, researchers have found that restoring certain aspects of this chromatin structure toward more youthful configurations can lead to measurable reversals in various signs of aging. Things like improvements in tissue function and shifts in biological age markers toward more youthful patterns. This represents one of the more concrete pieces of evidence supporting this broader idea of epigenetic reversibility. It's not simply a theoretical possibility. In animal models, at least, it's something that has been demonstrated experimentally. Now, let's turn to another area of active research. One that connects back to something we explored earlier in this very chapter, the cardiovascular system and the role of blood vessels in delivering what tissues need to function. Vascular rejuvenation improves tissue function in experiments. Researchers have explored various approaches aimed specifically at the blood vessels themselves, approaches designed to restore some of the flexibility and responsiveness that tends to decline with age, as we discussed when exploring arterial stiffness. And in experimental studies, when researchers have successfully achieved some degree of vascular rejuvenation, restoring more youthful vascular function, they've observed downstream improvements in the tissues that depend on that blood supply. This makes a great deal of sense when you consider everything we've explored throughout this journey. If vascular aging reduces tissue perfusion, as we discussed earlier, then improving vascular function might, in principle, support the health of tissues throughout the body that rely on that blood flow. It's another example of how interconnected these systems truly are, and how addressing one underlying mechanism might ripple outward with benefits across multiple systems simultaneously. Now, as we bring this chapter toward its close, let's step back and consider the broader implications of everything we've explored. Throughout this entire journey, we've described aging as an extraordinarily complex, deeply interconnected process. Hundreds of mechanisms woven together across every system of the body. And yet, increasingly, researchers are beginning to ask a rather different kind of question. What if aging could be approached not simply as an inevitable background process, but as something that could be medically addressed? Aging may be treatable as a medical condition. This represents a genuinely significant shift in perspective. For most of medical history, diseases have been treated individually. Heart disease, treated separately from arthritis, separately from cognitive decline, separately from each of the many other conditions that tend to emerge with age. But if, as we discussed earlier, many of these conditions share common underlying mechanisms, cellular senescence, mitochondrial dysfunction, epigenetic drift, chronic inflammation, then, perhaps, addressing these shared mechanisms directly could influence multiple conditions at once. This doesn't mean aging would simply be cured, but it does suggest a shift toward addressing the underlying process rather than only its many downstream consequences. And achieving this kind of shift requires something rather significant in terms of how research itself is conducted. Aging research is now interdisciplinary. This field draws together researchers from an extraordinary range of backgrounds. molecular biologists studying cellular mechanisms, geneticists exploring genetic influences, clinicians working directly with patients, data scientists developing and refining biomarkers of aging, and many others besides. This kind of interdisciplinary collaboration reflects the nature of aging itself. As we've seen throughout this entire journey, aging isn't a single process confined to a single field of study. It touches genetics, cell biology, immunology, cardiology, neuroscience, nutrition, psychology, and so much more. And given this complexity, it's perhaps unsurprising that researchers increasingly believe the future of aging interventions won't rely on any single approach. Combination therapies are likely the future approach. Rather than a single drug or a single intervention designed to fix aging entirely, researchers increasingly anticipate that effective approaches will involve combinations of different interventions, perhaps senolytics, alongside approaches that support mitochondrial function, perhaps lifestyle interventions like exercise and dietary approaches alongside more targeted molecular therapies. This makes sense when you consider everything we've explored. If aging involves many interconnected mechanisms, then addressing only one of those mechanisms in isolation may have limited effects. But addressing several simultaneously may have effects that are more than simply additive effects that reflect the deeply interconnected nature of the underlying biology itself. And so, we arrive at the final point of this chapter, a point that, in many ways, brings together everything we've explored throughout this entire journey. Evidence suggests lifespan can be extended beyond current limits. This is, perhaps, one of the more profound implications of everything we've discussed. For most of human history, lifespan has followed certain broad patterns that, while varying somewhat between individuals and populations, have generally remained within certain recognizable limits. But the research we've explored throughout this chapter, senolytics, xenomorphics, cellular reprogramming, epigenetic restoration, vascular rejuvenation, suggests something rather different. It suggests that these limits may not be as fixed as once believed. This doesn't mean dramatic increases in lifespan are imminent or guaranteed. The path from laboratory findings in animal models to safe and effective applications in humans is often long and complex. But the underlying science, the idea that aging is biologically plastic, that its rate can be modified, that many of its underlying mechanisms can be influenced and, in some cases, even partially reversed, represents a genuinely new chapter in how we understand this most universal of human experiences. And so, as we draw this chapter, and in many ways, this entire journey toward its close, let's take a moment to reflect on everything we've explored together. We began with a question. What does it actually mean for something to grow old? And from there, we journeyed through an extraordinary landscape. Through epigenetic clocks and the hidden molecular signatures of biological age. Through the quiet, relentless accumulation of cellular damage. DNA repaired 100,000 times a day. Through mitochondria and sirtuins and the ancient dormant echoes of endogenous retroviruses. We explored how aging shows up in muscles that begin their quiet decline in our 30s, in bones, in joints, in arteries, and hearts, working just a little harder with each passing year. We explored the senses, taste and smell, and hearing and sight, and the vast, invisible ecosystems within our own gut. And then we turned to something more personal. We discovered that genetics explains only a small fraction of how we age. That lifestyle, environment, connection, resilience, and purpose shape this process far more profoundly than many of us ever realized. We explored the U-shaped curve of happiness, the quiet, unexpected gifts of later life. And finally, we arrived here at the frontier of modern science, senolytics, reprogramming, chromatin restoration, and the genuinely remarkable possibility that the long, slow story of human aging may not be as fixed as we once believed. Aging is not a single thread. It is a vast, intricate, endlessly interconnected tapestry woven from cells and tissues, from genes and environments, from habits and choices, and from the quiet, immeasurable influence of the lives we live each and every day. And whatever the future may hold for this remarkable field of science, one thing remains true. The story of aging is the story of being alive. And it is a story still being written.